So the topic is what's the evidence that remyelination occurs. So I have about 5 slides about sort of this specific issue, but I thought I'd give an introduction and some further comments about remyelination as well. But I'll start out with an image that I brought along for you. So any suggestion, not by the team, but by our guests, what might unify these two animals? On the left, we've got a frog. What's the other one? What do you think? Squid, yes, a giant squid. And which physiological property do they share? Well, their conduction velocity is identical, so about 25 meters per second. And that, although the frog's axon is actually 30 times smaller in diameter than the giant squid. So that gives a sort of little indication of why myelination is actually good for the frog and for us as mammals or humans. Because what myelin actually does is written up here. It is responsible or part of saltatory conduction. That means in a normal, sort of in an unmyelinated nerve fiber, the impulse has to propagate from each sort of sodium channel to the next, okay, and that takes much longer than when you have a myelin sheath and the impulse is actually jumps from this node, called node of Ranvier, to the next node. It works like a sort of insulator around nerve fibers. So that has 2 effects. One is the acceleration of the propagation of the impulse, and it's also much more energy efficient. Just to show you a real image of this is an electron microscopic image of an axon here with this nucleus, and these are all, this is not one big pack, but it's all sort of individual little layers, like in many layers wrapped around an axon, and it's actually up to 100 times that one axon can be actually wrapped by myelin layers. And that has a huge effect, even within us humans. Up here, these are sort of motor fibers, so they have this type of conduction velocity when they are myelinated. And down here, these are sort of pain conducting fibers, and they're much slower than their myelinated motor colleagues or correspondents. Okay, taking it to multiple sclerosis, I brought you an image of a scan. Many of you certainly will have had one of those, and it's probably quite straightforward to imagine that these lesions here have sort of reflect changes here on this post-mortem section of somebody who died with MS when you stain this section for myelin. So all that's dark here, okay, is myelinated, and here this brighter aspect is the cortical ribbon, and in here we've got these white spots that correspond to these MR lesions. So this is demyelination. And when you look a bit closer in the microscope, you can see something like this. Don't look at this down here. This is the cortex, but there's one and another demyelinating lesion, quite characteristically with a central blood vessel here. And it's only sort of by chance that this one was kind of hit tangentially, so you can actually follow the blood vessel all the way through. All the blue stuff here is so-called Luxol fast blue, staining for myelin, and myelin is lost here and there. Okay, moving from here. So what is remyelination when we're talking about demyelination, and what is it good for? Well, remyelinate or else. These are the 2 basic pathways that a lesion can go after a bout of demyelination. So these are neuron cell bodies, and they're axons here in the center. You have these myelin sheaths around it with their Ranvier changes. And one oligodendrocyte, which is the cell type that produces myelin, usually myelinates several nerve fibers. Now, once this area here is demyelinated and the oligodendrocyte is gone, or many of them, two things can happen. Either there's something happening, as in a repair kicks in, which is here, this pathway, remyelination. And as you can already see, these myelin sheaths are thinner, and they have sort of shorter internodes, now remember the word, internodes, than the original ones. Or you can have this situation, which we obviously will try to avoid, which is axonal degeneration. So the nerve fiber that was previously myelinated here has lost sort of its support by the myelin and degenerates, okay? And that is what we believe is very much sort of the pathological correlate of disease progression. This is some more images showing up here myelinated fibers, just sort of transversely sort of cut. So you have here in the center the nerve fiber and the myelin layer around. Okay, everything nice and blue. Here is demyelination, all pretty white except for a few blue spots, and then remyelination. Here, all this is now from an experimental setting, and you have these thinner layers of myelin around here. This is just another sketch that illustrates this point. Now, we think that remyelination not only it doesn't only sort of protect axons from degenerating, but there's clear evidence that there's also functional recovery. So this is not only sort of showing off and sort of wrapping a useless new layer around, but it actually does work, certainly in the experimental setting. Okay, does it occur in MS? These were all sort of examples from experimental settings. And these are the 5 slides that Gavin had actually asked me for. The evidence for remyelination in MS. Well, this is an example of our own work. Clearly there's here this demyelinated area, and I don't know whether that projects well in the back, but under the light microscope, such an area here is very characteristic of remyelination. When you look a bit closer, I didn't bring the sort of the closer or higher magnification here, but you can see that there are more thinly myelinated fibers than in the surrounding normal white matter. So this is multiple sclerosis histology. Another example, these were two brains, two brains of people who died with MS, and the authors here analyzed 168 lesions, and despite these the people who had died with MS, having had the disease for over 20 years, they found these areas, i.e. remyelinated areas, in about 123 of their 168 lesions. So it's quite successful sort of remyelinating capacity, at least in these two brains. Now you could say, well, this is only 2 cases. Let's have a look at a larger case series. And this is what you can see here. This is 51, and from a group based in Vienna. And what is interesting here is that remyelination was extensive in a subset, as they say. So now what is this subset? A subset of patients where there was a lot of remyelination were those with relapsing or remitting MS. About half of them had lesions, in 60 to 100% of their lesions, they had signs of remyelination, and patients with primary progressive MS, whereas very few with secondary progressive MS. So suggesting that remyelination is something that happens more likely earlier on in the disease. And yeah, the failure of that may actually underlie disease progression. Now, sorry, I should have perhaps announced this, but it's we do have a collaboration with the MS Society's tissue bank, and this is sort of the source from where we receive our tissue. So this is one sample of a brain from one hemisphere, unfixed here in this situation. And what you can see here are these opaque changes, and in the so-called white matter here, in fact, it is white, not as it looks on MRI scans. Usually, it's very much darker than the actual gray matter. So here it is white, and you see here these lesions. But it's not only the white matter, but also the gray matter. When you look closely, you can see even in the gray matter there are lesions of demyelination. And now the big question is, of course, or was, whether there's also remyelination, so this repair process going on in the gray matter. And in fact, there's been a paper on quite a large sample showing that there's extensive also cortical, so gray matter, remyelinations, not only in the white matter. It's illustrated here. On this side, you see normal myelin. This is cortex without myelin, so it's quite a contrast here. This is the normal control brain, and here there's a lesion again in the cortex, but with a few patches of thinly myelinated or irregular myelin around. So what is the next question I thought to ask is, what is good and what is bad for remyelination to occur? And I'm going just briefly back to the earlier sketch, that we basically have these two pathways and what determines them. So we have that little sketch up here with the corresponding brain. So when we start here with normal white matter, we have quite a number of different cell types in here. What is of particular interest for us are these cells. These are the oligodendrocytes, so sitting here, myelinating away nerve fibers, and a group of cells that you will notice on the next sketch, which are so-called precursor cells. One could call them stem cells, but precursor cells is probably more appropriate. So now, when demyelination occurs, obviously oligodendrocytes and myelin is being sort of destroyed, and that's part of the inflammatory response. And what happens subsequently, or almost in conjunction with it, it's not that it's first demyelinated and then there's a gap and then remyelinating starts. It's probably really something that goes hand in hand. It's sort of immediately starting to repair. And so these particularly these other little cells, microglia and astrocytes, they start producing factors that activate these precursor cells. Precursor cells are important because once your oligodendrocytes are dead, you need new ones to produce new oligodendrocytes to then be able to remyelinate. Okay, and this is what is illustrated here. This is the recruitment phase of these precursor cells back into the area that was attacked earlier on. Okay, so it's repopulated. So there's a recruitment phase, and then there's a differentiation phase. That means the oligodendrocyte precursor cell needs to differentiate to a full-blown oligodendrocyte that can then do its job and remyelinate this lesion. Now, why does it fail in MS? And as I said, there are two phases. recruitment and differentiation. Both phases can be hampered. Well, when it comes to recruitment, there can be too few of these cells. That can sometimes be the case, particularly in large lesions, that there's a lack of guidance cues. Actually, for example, there's a group of signaling molecules called the semaphorin that are important here. Or the differentiation, so the maturation of oligodendrocyte precursor cells is disturbed. And most of the experimental evidence seems to suggest that it's actually a differentiation that is more important than recruitment. So cells are available or there, or precursor cells are there, but differentiation is not happening, and therefore they're not really doing their job. So there are the presence of either negative factors or positive, or the absence of positive factors may be responsible here. If remyelination is happening, and we'd like to observe this and perhaps even produce or create a drug that fosters this process, it'd be useful to actually know it happens in somebody with MS. And as we all know, the course of MS is highly variable, so to sort of look for clinical markers is often time-consuming. It takes a long time. So with using tools to observe remyelination without having to do sort of long-term follow-ups, clinical follow-ups are warranted or needed. And one of them is actually an MRI marker. It's work that we've been involved in ourselves. This is based on an effect in MRI called magnetization transfer. The problem is in MRI that you're basically looking at protons, so water molecules. However, what you're really interested in are the protons that are incorporated or part of macromolecules. And these, for physics reasons, their signal dies down very quickly. So we use a trick, which is magnetization transfer, to observe the proportion of protons bound to macromolecules, and that technique is magnetization transfer ratio. And what we've shown here is, again, in post-mortem brain tissue, that there's actually a very good correlation between this MRI marker and the proportion of myelin, or the amount of myelin in the brain. So it's quite a reasonable predictor for that. We can obviously do better, and when you use a high-powered machine, you can actually visualize this rim of remyelination. So this is a lesion here, okay, and this is this half moon of remyelinated area that corresponds directly to this area on the MRI scan. But we're not quite there yet. The highest field scanners currently in use are 7 Tesla, and we've got only one in the UK so far, which is in Nottingham. So it's still a little bit of future task. Finally, can we actually help the nervous system to remyelinate? And there are various strategies, including those mentioned here. LINGO is up here, it's written out down there. It's one of the inhibiting factors that I mentioned that are inhibiting differentiation. There's a phase 1 trial currently underway by Biogen, I think it's in Switzerland, I believe. There's you also find actually candidates that are currently already in use, but which are not sort of fully sort of investigated for their potential to remyelinate. And there's been one paper actually by our group, Sam Jackson and David Baker and Gavin have been involved in this, showing that fingolimod does have a sort of regenerative potential apart from its primary or better known mechanism. Natalizumab, obviously well known to all of you. There's currently a study using magnetization transfer to look whether there's indications for remyelination. And the most recent sort of kid on the block, but in very early, certainly experimental phase, the RXR signaling pathway. So that is that. Any questions, comments?